it's a great pleasure to welcome today's speaker, Professor Paul Hertzog. Paul is the Director of the Centre for Innate Immunity and Infectious Diseases at the Monash Institute of Medical Research. He's a long-time colleague of many of us at the Institute, particularly in relation to the SOX proteins. It's always a pleasure to hear what he's been up to. He did his PhD here in Melbourne and then did 2 postdoc positions, one in the US and one in the UK, and has made a career out of studying the interferons, particularly in the context of innate immune signalling and signal transduction. He's had a couple of really fascinating stories emerge over the last couple of years. They were published last year, I believe, in Science and Nature Immunology, and I'm sure that he's going to tell us about them today. Thanks, Paul.
Indeed. Thanks, Sandra. Thank you very much. It's a great pleasure to come here for a seminar at the Wednesday series. And as Sandra said, especially at WEHI, where I've had some really productive collaborations and made a lot of good friends over the years. No surprise to you that I'll be talking about interferons, which I've spent decades working on. And indeed, it's a fascinating field that's been going for over 60 years now. It's even from the very early days in the 60s, it was a component of potions, really, that they were putting into people to treat certain cancers. The signalling pathways have given us the JAK-STAT pathway, which the SOX proteins target that Sandra mentions that were co-discovered here. And really, it's a system where a lot of elements of gene regulation have been worked out with Tom Maniatis and the beta promoter and so forth. So both in terms of a cytokine, a biologically relevant molecule, and a system that can tell us some general things about the biological systems in general, it's been fascinating, and it's those elements that keep me interested. So what I'll talk about today are a few things, but I thought I'd just say why the title. So, you know, why selective regulation? And I'll talk about 3 elements of this in today's talk. So when we talk about type 1 interferons, which we will be, it's important to remember that this is one of a gene locus that contains about 17 different genes encoding different members of these protein family. They're essentially never produced together, but often you get multiples of them produced in different amounts. These alphas or beta interferons in turn tend to work through a common receptor, we think. Activates the JAK-STAT pathways. I hope no one gets seasick because there's about 50 animations on this slide. And these diverse pathways then activate their gene programs that basically encode effector proteins that develop the response. Now, the various genes here we've collected, and I talked about this in an undergraduate lecture, so I'm not going to cover this topic much, but in this database, we collected every data set we could find of a cell, an animal, a human being treated with interferon, and put them in there and tried to annotate pathways in there and to have a sort of definition of what it is. So depending on your cutoff, there are about 3,000 interferon-regulated genes. If you make it statistically significant, it's probably up around 4,000 or 5,000 genes. However, depending on the circumstance, if it's a particular cell type, if it's in a situation where you're getting inhibition by SOX or other proteins, viral products, the thing is you only get certain of these pathways activated. You never get that whole repertoire. And, you know, so you may end up with some hundreds of interferon genes regulated, and these are going to be responsible then for encoding proteins that drive antiviral responses, modulate cell survival, activation and proliferation, and so forth. So these effects then, depending on the gene repertoire, will vary from cell type and different circumstances, whether it be a cancer or infection. So the questions around selective regulation are, I mean, what determines which interferons are produced in different circumstances? I mean, do they really all interact the same with their receptors? Is this a very redundant system? And how are some of these pathways regulated? So I'll talk about these 3 aspects today. So sometime on the rules regulating the expression of this cytokine family, some elements of how interferons or different interferons might work differently in their receptor interaction, and some new data, time permitting, on how this system is regulated, particularly at the gene expression level. So firstly about the interferon. So as I said, the locus, which is on 9p in humans, chromosome 4 in mice, contains multiple genes. The black arrows there are protein-coding genes, the others are pseudogenes, so it's a snapshot in time of a very actively evolving gene locus where genes are coming in and out of existence. There are interferon alphas, about 14 of them there in humans and mice, interferon beta at one end of the locus, epsilon at the other end, and I'll talk about these. Interferon omegas and interferon kappas, which we don't hear about that much. So these type 1 proteins are very similar. You can see down the right-hand side here, the family of alphas are about 75 to 100% identical. There are in fact 2 different genes encoding identical polypeptides. The genes are very simple, 660 or so nucleotides, no introns, small proximal promoters. And all of these interferons, including the more diverse beta interferons, which are about 25% identical, seem to act through this same heterodimeric receptor complex, activate JAK-STAT signaling as well as other pathways, and activate then these characteristic biological responses. So what we do know about the divergence of the proteins is most of them seem to activate the same biological responses. They do it, however, though, with different affinities and effectiveness, particularly at least amongst the different subtypes of alpha interferons. So you could ask, you know, why have so many? Why evolve a system that operates like this? Is there a lot of functional redundancy? And certainly, as Sandra said, you know, I work on interferons in the context of the innate immune response. All these innate protein sensors and receptors that have been discovered over the last 15 or so years all seem to drive an interferon response. We know it's important because viruses target it, and without that targeting, their long-term survival and chronicity are challenged. 20 years now of working on IFNAR knockout mice have shown us that this part of the system is important in all sorts of systems. Okay, so basically some of the general rules then, some data and facts and some propositions from me is, of all these interferons, you never get all of them made. You rarely, probably with the exception of a couple I'll talk about today, get one of them. So they're generally made in small subsets, induced by pathogens, both viral and bacterial, also induced by commensals, so lactobacillus stimulates huge levels of interferons, and also induced by danger-associated molecular patterns that often seem to mimic infections by generating DNA or other nucleic acids from dying cells. So a diverse range of stimuli. If we look at the promoters of these genes as opposed to the protein coding sequences, I think we also get some answers as to their existence. So there is a core set here that are often made together that have very similar looking promoters. They certainly have the IRF7 and other sites that constitute the viral response element that's been well characterized. Others, such as beta, is the only one with an NF-kappa B site. It makes it the only one made in response to LPS. These two up here, alpha 1 and 13, are the ones that encode identical polypeptides. You can see the binding sites in their promoters are different, so they're going to be made in different times and in different responses. So you could speculate then that at least amongst the alpha interferons, the diversity of promoters here are one of the important reasons why you have so many subtypes. And maybe what they do is present a diversity of targets for these various upstream pathways that need to drive interferon in order to elicit effective innate host responses. And this is certainly consistent with if you look at the phylogeny of these interferons, and basically you're not really meant to read it, these are speciations, so whether it's cat or human or mouse, other mammals and species, you seem to immediately get the ability to make multiple interferon alpha subtypes, sort of shown here by mouse and human interferons, where the components are more similar to each other than they are across species. So what you don't get is a mouse interferon more similar to a human interferon subtype than others, which you might expect such a pattern if you are conserving a specific function of each gene. What it seems to be important is you get the repertoire. That, however, so this is consistent with that hypothesis for the part of the explanation for the existence of multiple alpha subtypes. Not true, however, for beta and epsilon, which I'll talk about today, where the human and mouse are more similar than they are to other members of their species type 1 interferon family, inferring that these may indeed have evolved different functions or specific functions. Okay, so the second part then of considering why we have so many interferons and rules and regulations that govern which ones are produced at which time will take me to a short discussion of part 1 of the talk on interferon epsilon. So this is what we thought a type 1 interferon, a gene we noticed an embarrassingly long time ago. It took us nearly 10 years to characterise this. It was done by Matt Hardy, who's now at CSL, who was a student of mine. So this interferon is part of the type 1 locus. And we wanted then to test whether it had typical properties of the type 1 interferon. So they typically are defined by the fact that they work through these two IFNAR1 and IFNAR2 components of a receptor. They tend to activate signature interferon-regulated genes, generally inducible by pattern recognition receptors. They can protect against viral infection. Depending on the nature of a bacterial infection or the immune response required to combat it, interferons can either be pro or anti a bacterial infection. They can be pro-inflammatory and regulate many of the immune effector cells. So this interferon then, in short, when we found it, is about 30% identity to a consensus alpha sequence or to beta, so it's quite different. Modelling would have predicted it folds up like a typical 4-alpha helical cytokine like the other interferons and many cytokines indeed. After a lot of trouble, this was a difficult protein to make and stabilise, unlike the other type 1 interferons, which are the hardiest proteins you can make. They're acid and heat stable. This seems to be a very unstable protein. We made it, as I'll show you later, we made antibodies to it. We showed that in cells, this is data from bone marrow-derived macrophages. It induces some classic ISGs like ISG15, IRF7 and 25OAS. These are antiviral genes. In the two cells from either of the IFNAR1 or IFNAR2 knockouts, there's no induction. So this seems to indicate that it is a true type 1 interferon because it requires each of those receptors for its signalling. So next then we looked at whether it was inducible by pattern recognition pathways, and this is an example of some of the data we have. The yellow blocks here represent interferon epsilon. It's measured by RT-PCR. This is again in macrophages. The positive controls are measuring beta or IL-6 there, induced by a variety of TLR ligands. We've done all the TLR ligands, most pattern recognition receptors, a number of hematopoietic cells and epithelial cell lines again. In none of these circumstances do we get any induction of interferon epsilon. And in fact, if we make a promoter reporter and co-transfect with the main IRFs 3, 5 and 7 that drive other type 1 interferon genes or reporter constructs, again, there's no driving of this interferon. So it's looking incredibly different. Not surprising, because if you look at this very informative data on the interferon epsilon promoter, you see it has none of the typical sites we'd see in the other type 1 interferons like STAT sites, NF-kappa B in the case of beta, or IRF sites. So when is it expressed? We did a RT-PCR scan of a variety of tissues and found, like interferon beta or alpha in most tissues, there's no detectable expression, with the very notable exception of the female reproductive tract. So this is constitutively expressed at very high levels in the uterus and to a lesser extent in the ovary. Given that location then, we looked at to see what cells produced it. You can see there it's produced by the endometrial epithelial cells. Clean data in the knockout there showing the specificity of the antibody and the reactivity. So this is again quite unlike other type 1 interferons, which, while they can be produced by a wide number of cells, usually we associate with hematopoietic cells. We wanted to know whether it was hormonally regulated given its location, and certainly it is. At different stages of the estrous cycle, the levels range about 40-fold from the highest at estrus, lowest at diestrus or proestrus. It also fluctuates during pregnancy, so compared to the normal levels you see, it's down-regulated in pregnancy, lowest or almost completely undetectable at the date 4.5 in the mouse, which is the point of implantation. It's also not detectable in pseudo-pregnant mice. So these are mice that are mated with vasectomized males. They don't have the physical presence of the embryo, but they do have the hormonal changes consistent with pregnancy, implying that it's a hormonally regulated cytokine. Indeed, if we ovariectomize mice and come back and treat them with estrogen, you get a stimulation of epsilon level. If we treat mice with progesterone or progesterone derivatives, you get a suppression of epsilon production, which is consistent with these patterns you see during the estrous cycle or during pregnancy. So again, this is really different to the other type 1 interferons, which are not hormonally regulated. We wanted to see if the same was true in the human as the mouse, and at least on a gross level at a tissue panel, it certainly is most highly expressed in the endometrium. The expression, again, is proportional to the levels of estrogen, so it's highest in the proliferative stage, lower in the secretory stage of the menstrual cycle, and very difficult to detect in postmenstrual women. Also, the recombinant protein that we make drives similar ISGs, shown here for 2,5-OAS and the MX protein. So, at least as far as we can look superficially for the moment, the same sort of rules seem to apply for the human and the mouse. So what's regulating this expression? These are some of the elements that you can get by a transfact or such prediction of the promoter. The most notable to us, probably because I have a history of working on it, was the very highly conserved ETS binding sites. These are transcription factors, ETS 1 and 2, that are often associated with hematopoiesis. There is also a subclass of ETS-like factors that are involved in branching morphogenesis of epithelial organs. So we looked to see whether the ETS sites might be regulating, and indeed it did. If we again take the promoter reporter, co-transfect with a number of ETS family members, we show that these can indeed drive the expression. There are 2 ETS sites. In fact, we showed that it's the more proximal one, that's the active one by deletion constructs. And of the ETS factors, the epithelial located ALF3 was the most active, and indeed this is co-located, co-expressed in the endometrium. We did point mutations of the predicted ETS site to show that that it was indeed responsible for driving expression. So this is very interesting because ALF3 is a constitutive gene in epithelium, including the uterus, as I said, and often involved in developmental abnormalities when it's deleted in knockout mice. We've also shown that the expression of ALF3 parallels, at least during pregnancy, the expression of interferon epsilon, consistent with it being a main driver of this cytokine, and perhaps implying an indirect mechanism of regulation by the hormones. And that's something we're still trying to come to grips with, exactly how this mechanism occurs. We don't actually see obvious hormone receptor sites in the interferon epsilon promoter, but they are difficult to predict. So then to look more at the function, we generated some knockout mice. We started conditional, ended up we didn't need them because these mice are born and develop very normally. With very extensive studies with data I won't show you, both male and females have been shown to be fertile over multiple rounds of pregnancies as well. So it seems that the constitutive expression has no, or at least its absence has no obvious effect on fertility or development. So we looked at it then at some models of infection. The first one we examined was herpes simplex infection, HSV2 infection of mice. You can see by clinical score the knockout mice have significantly worse disease. That should be seen. There is actually a significant loss of body weight after 7 days of infection. If you look at viral quantitation early or later during the infection, at 3 days or 7 days, you can see a greater yield of virus, both at the early stages and the later stage, and this is true in the spinal cord and brainstem because, as you would know, this is an ascending infection that descends from the reproductive tract to the brainstem. So this shows a very important and significant effect of interferon epsilon in protecting against HSV2 infection. The effect, I should say, is probably not as dramatic you see with the receptor-deficient mice, and probably that's because the receptor-deficient mice will be looking at a sum of this constitutive interferon and the other type 1 interferons which will be induced during this infection. The next infection we looked at was a bacterial infection using Chlamydia muridarum in collaboration with Phil Hansborough in Newcastle. Again, in the light grey bars, you can see a clinical score of the disease, which is much worse in the knockout mice over the 30-day course of this infection. Again, if you look at the bottom two graphs, you can see both early infection. At 3 days after infection, there's a significantly higher number of bacteria recovered in the knockout mice. And if you look at the later stage of infection, 30 days post-infection, when normal mice have cleared the bacterium, you can see very, very significant levels of this bacteria in the knockout mice, implying that it's important both in the initiation stage of the infection and in the clearance phase as well. It's also interesting and worth noting that interferon epsilon seems to be the only type 1 interferon that protects against Chlamydia muridarum. So earlier studies that have been published by others on the receptor-deficient mice implied that type 1 interferons actually made the disease worse. And by using adoptive transfer studies, they show that CD8 cells, which along with NK cells are important in controlling this infection, were actually activated by type 1 interferons to drive the inflammation and make the disease worse. So here we have a very counteractive effect of one type 1 interferon and another, perhaps by virtue of the compartments they're secreted into, but that's something we remain working on. So as I said, NK cells are one of the effector cells that are important in clearing this disease. Consistent with that, the knockout mice show lower levels of these cells. We've shown if you administer intravaginally some recombinant protein, you can restore these levels back up, and at the same time then you get a dose-dependent decrease in the yield of bacteria, showing that the recombinant protein is active in vivo and may even have therapeutic potential against infections of the reproductive tract. So the mechanism of this is somewhat difficult to come to grips with. I find it difficult to conjure in my own mind compared to the inducible cytokines we're all used to dealing with, how this in fact works. So certainly we've measured it during those infections. We measured it's there before the infection, it's there during it, it's there afterwards, and apart from some minor hormonal fluctuations, doesn't seem to change throughout the course of infection. If you look at some of the interferon-regulated genes, and some of these I should have pointed out at the start are important upstream effectors like TLR receptors, IRF7, which is a transcription factor which drives interferon itself. The levels of these IRGs are much lower and virtually immeasurable in the knockout mice, showing that the presence of epsilon, which is the only interferon we can detect, is important for priming some of these early response genes. And maybe that is the essence and at the heart of the mechanism where this works, to prime the system up so that it's ready to respond by the more familiar mechanisms. Okay, so to conclude on this interferon then, interferon epsilon is a new type 1 interferon cytokine. It has very unique regulatory properties. It's constitutive, it's hormonally regulated, not regulated by pathogens, and it's most highly expressed in the female reproductive tract, although recent data would say probably not exclusively. Not required for physiological functions, it seems, but critical for protection against both bacterial and viral infections. So in terms of the broader concept of thinking about why we have so many type 1 interferons, this appears to be, colloquially anyway, a designer type 1 interferon that we've evolved to regulate immune response in the female reproductive tract particularly. And this is a location, remember, that must be protected from infection, must function amid massive fluxes of cells and hormones, and yet still be tolerant to embryo implantation and development. So we're interested in this in the context of viral infections and what importance it might be. So if you consider a classic type 1 interferon compared to epsilon, this is pathogen-driven through familiar pathways, drives JAK-STAT pathway and IRGs, and for example, are antiviral effectors. Epsilon is not regulated by that pathway, but by a hormone, seems to work through the same receptor, similar pathways, and similar antiviral effector molecules. So this one is transient and in high levels. The epsilon is sustained and fluctuating at probably low levels as far as we can tell. They're both have antiviral direct properties and can activate immune effector cells. I haven't shown you that data, but in vitro that's at least true. So if we think of some of the applications of these pathways, some of the vaccine adjuvants that are used do so because they work through pattern recognition receptors and drive interferon. Some of their effects are not so great in the absence of a type 1 interferon receptor, so this system is important. So those adjuvants are not going to affect this pathway. A lot of viruses have evolved inhibitors of interferon responses. Many of them work at the level of IRFs and driving interferon production. Again, they will not block the production of this interferon. So that's quite handy for a local protection agent. And the other element is that estrogen, this side is hormone regulated, both estrogen driven and progesterone driven. We have some collaborators interested now in looking at particularly this suppression by progesterone, because there's reasonable data out there inferring that women, particularly in places like sub-Saharan Africa that are on very highly susceptible to HIV infection and are on progesterone-based oral contraceptives, are more susceptible to some of these infections and STIs. And one proposition may be that this system is important in regulating that. So that's one project we'll be looking at in the future. Okay, I'll leave that story there and move on to the second part. And while staying a little bit on the topic of, you know, when do we get, what situations do we get different interferons on, but moving in the case of interferon beta, a more familiar type 1 interferon, to how it interacts with the receptor. So certainly this is another interferon that is made in different circumstances. As I mentioned, it has an NF-kappa B site in its promoter that the others don't. It has AP1 sites in the promoter. So not only is it pathogen-driven, but it's driven by MCSF during macrophage differentiation, by rank ligand during osteoclast differentiation, and has anti-proliferative effects that regulate some of those cell types during their genesis. So we've always known that, and by implications, the fact that it's made in different circumstances, in vitro, some of its actions are more potent than alphas or other type 1 interferons if you measure them. And for really unknown reasons, it's the only type 1 that has worked and is still used clinically in multiple sclerosis. So all of this infers that beta will have a different function to alphas, but we've never really had a mechanistic basis for doing that. So to just remind you of the way type 1 interferons are supposed to interact with their receptors, and this is similar to most other cytokines. There's usually a primary binding chain, in this case IFNAR2, which binds with moderate affinity. Once that's bound, it brings in a second signaling receptor, which, at least for alpha interferons, I'll show you different for others, has no ability to bind this IFNAR1 chain on its own. And this trimeric complex is what binds with high picomolar affinity and transmits signal. So we were looking at type 1 interferons, embarking a long time ago, championed this work by Nikki DeWeerd in my lab to do some crystallography studies, and we started making the receptor extracellular domains and ligands and incubating them. And this result turned up, and basically what it showed was that IFNAR1, this is run on native gels at the top, STS page at the bottom. You can show here, this is the receptor extracellular domain. So this data showed that interferon beta interacted and formed a complex with IFNAR1. So I said to the guys who were doing that at the time, that's wrong, go away and get the right result. There must be something wrong with the conditions, and they did it, and indeed it was right. Not only is it right, but it's a very stable complex. You can isolate it by ion exchange chromatography and gel filtration chromatography. It binds, in fact, with a measurable affinity by SPR measurements. Nikki then went on, and basically it binds at a level that you really don't see with interferon alphas. So the important thing is to emphasise that this is a beta IFNAR1 specific interaction that the alphas that we've tried do not do. She then went on to crystallise this protein in collaboration with guys in Jamie Ross-John's group at Monash to solve the structure of this interaction. You can see here the interferon beta sitting. The IFNAR1 has 4 subdomains. It basically interacts with the 3 of them that are at the N-terminal. The one that's membrane proximal, SD4, has minimal interactions, maybe a small interaction here. About 6 months before she actually got this crystal, a paper came out from by Thomas et al. Showing the structure of IFNAR1 and IFNAR2 bound to mutant interferon alphas in the human systems, which was a worry at the time, but very nice for Nikki that we got this alternate structure out. So this maps the predicted binding sites of the different domains on the ligand. You can see there's extensive interactions with the SD1, SD2 and SD3 domains of the receptor, very little with the other domain. And essentially, if you calculate the surface area bound, you get a very high interaction surface area compared to what you would get with another type 1 interferon. This data comes from the Thomas paper that shows a human type 1 interferon. I don't know why they used omega, maybe it was the only one that crystallized. But anyway, we get significantly higher interaction with beta, which probably explains why the affinity for that interaction is higher than the others. You can, of course, with these structures, identify important interacting residues on both the ligand and the receptor, and Nick is in the process of examining these and identifying the important ones at the moment. And you can certainly, if you line up the sequence, identify ones that seem to exist in beta that don't exist in the alphas that can become some of the prime candidates. However, the most important really question for us was, okay, this interaction happens. I mean, is it relevant in vivo and can it transduce a signal? The first experiments we did were to look at stat phosphorylation. This is by phosphor flow. And we used for this IFNAR2 null mice. So I've got in the bottom corner that just to remind people that they will still express IFNAR1, which is green. And if we add interferon beta, this unusual interaction, which could still occur on these cells, we can use that as a system to test whether or not it signals. So anyway, as you can see, in wild-type cells, in the IFNAR2 nulls, there is no sign of any signalling happening, whereas in the wild-type ones you get good stat phosphorylation, same in the IFNAR1 knockouts. If we look at some classic interferon-regulated genes, again, the wild-types induce, both the IFNAR1 and IFNAR2 nulls don't. So there seem to be this is adding beta to this cell. So there seemed to be no indication looking at classic signalling that anything was occurring. So we then looked just at the receptor level to see if we could see signs of internalization. This is basically before and after internalization in wild-type and IFNAR2 null cells. So it's slightly different and not as dramatic as you might see in wild-type cells, but there is some internalization, giving us an inkling that there may well then be some signals transmitted. So taking a global approach to this, we did microarray analysis. The system was using peritoneal elicited macrophages. These are just some plots to show the wild-type response. And you can see in the IFNAR2 response, there are both some genes activated and some genes repressed. The Venn diagram shows you the extent of this. While you get 3,000 odd genes induced in a wild-type mouse, most of these are not induced in the IFNAR2 mice treated with beta. So a very small subcomponent of them are, and there are some genes induced that you wouldn't normally see, but at least we had evidence of signalling occurring in these mice. We then went on and validated some genes that didn't induce in the wild types, but did in these. Some of these genes are quite interesting because they have been associated with inflammatory response to LPS. So we looked at a model of septic shock in mice where you administer quite high doses of LPS. The wild-type mice die, or most of them die, within 3 days. IFNAR1 mice have been published before to rescue this response, and in fact crosses with a lot of knockouts of many different components of the interferon system rescue this phenotype. Interferon beta does, IRF3 knockout mice does. So we've known that type 1 interferons are implied in this. But as you can see there, the IFNAR2 mice, which will still have this beta IFNAR1 signal, still basically die at a similar rate. And in fact, there is a subset of genes that you would have seen in that Venn diagram that are induced in common in the wild type and also in these knockout mice. So, and basically, again, some of these have been associated independently with the pathology of sepsis in this model. So, I mean, this provides data then that this signal can occur in in vivo from an interferon beta IFNAR1 and may be of some consequence. We still really have much to characterise in that response. But it's worth sort of thinking. So we've had, in fact, these IFNAR2 null mice for a while. We've published very little on them because they always showed a difference to the IFNAR1s. They showed 90% common phenotype and always an odd, quirky difference that we could not understand. But maybe this very recent data explains some of the differences we've seen. So some of these differences are interesting. One is, in fact, the SOX knockout mice, which were generated here. You will all be familiar with the lethality of the SOX1 knockout mice that die by 3 days of a multi-organ inflammatory disease. Warren and Robin Starr and Doug and Nick and co, Sandra, have published that this is an interferon-driven disease. You can rescue it with interferon gamma null mice, as you can see here, long-term. We also published in collaboration with people here that the type 1 interferon system could also rescue this. So if you cross them with the IFNAR1 knockout mice, you have pretty much 100% viability at the 3 weeks where the knockout mice essentially have 100% lethality, but the mice then do die later on. Similarly to some other phenotypes and multiple crosses on the SOX mice, which seem to be a complicated autoimmune disease. However, in this system, which we published many years ago, the IFNAR2 mice, again, do not rescue this. And maybe that is due to the fact that some of this inflammation is being driven by this beta-IFNAR1 interaction. And another situation where this is also true is work done quite a while ago with Connie Wong and Peter Crack, who's now over at Melbourne University, showing that in mice, if you do this mid-cerebral artery occlusion model of stroke, in brain slices, you can measure the size of the infarct. Here we showed that the IFNAR1 knockout mice have an amazing level of rescue of this. However, the IFNAR2 crosses don't alter it at all, and the phenotype is very much like this. Again, another situation. So I guess what I'm saying is that there seem to be many notions or examples of inflammatory diseases where perhaps this signalling axis is important. This is just to summarise this aspect of the work. It's a summary done in News and Views by Leon Plutanius and co. So essentially, the summary is that this is a really unusual interaction that interferon alpha can't engage in, and it's what would normally be known as the beta non-binding chain of the receptor. Seems to activate signals that you don't normally get, doesn't activate this classic JAK-STAT pathway, but does drive the expression of some genes. I mean, the other thing I would have to say is that this is probably an oversimplification of the mechanism of the interaction. There are very few, if any, examples to my knowledge of a single chain interaction like that would transmit a signal. So whether these may homodimerize, which we have no evidence of in vivo on cells or in a test tube, or whether there are other transmembrane components that have to come in and help this transmitter signal, are interesting things we have to pursue. I'd welcome feedback on this, of course, from experts in the audience. Okay, so in the last little bit, I'll just scoot through probably some recent data that we've done on a different aspect of regulating selectively different components of the interferon system. And most of this was done by an awesome PhD student, Sam Forster, who's now just gone for a postdoc at the Sanger Centre. So he's done both the wet lab and the bioinformatics components of this. So you'll remember this component. So we're still focused then on the interferon system. I've told you we've had an interest for a while in looking at these subsets, the interferome and other projects of trying to annotate some of these subsets, work out what the signalling pathways they are, so that when we see them, we might know the pathways that are activated. That's true of the selective regulation by SOX proteins of that unusual beta signalling I showed you, and yet to be determined whether epsilon may do it. However, as you know, there is another level of regulation of protein expression of these interferon-regulated genes, and that's by non-coding RNAs and microRNAs. So in order to get a handle on this, we decided to do an RNA-seq experiment of an interferon response, which has generated some really interesting data. And there are two things that you should remember from this talk. One is that's probably not expected that we've uncovered a huge array of both known and novel microRNAs that regulate these thousands of interferon-regulated genes, and they, I think, are potentially really important modulators of things like SOX1 that we've never been able to get a handle on modulating. The second thing is that we really didn't expect, and very much due to Sam's work, was that there's a lot of alterations and processings of RNA that are interferon-induced that we didn't expect to see, that I think, again, might have broader implications. So the experimental system we use is interferon beta treatment of bone marrow-derived macrophages, as a lot of you will probably use. This was done at a time when we had the solid next-gen sequencing system. We separated the samples into large and small RNA so we could get good coverage of these reads and did fairly standard sort of processing. So this shows typical data of the large RNA. So we did a time course over 12 hours. Most of the data we've processed is for 1 or 2 hours. So you typically get some genes are induced, you know, both at 1 or 3 hours. Some are later induced, some are earlier. They come and they go. The blue marks genes that are suppressed. So in summary, down the right-hand side here, what you can see is we discovered 1,500 protein-coding genes that are regulated, consistent with the numbers I showed you earlier. Some pseudogenes, transcribed pseudogenes, and importantly, precursor microRNAs that are, this is important because it becomes part of our ability to find them. SNO RNAs and a small number of link RNAs. So all of these are awesome projects that we'll never have time to pursue, probably. There are also large things we couldn't really define, large splice sequences, unspliced sequences, and small splice sequences that they're coded at the moment. However, the interesting thing that Sam noted was that in the stimulated conditions, you can see here in blue, is that there are a lot of unannotated sequences. So these couldn't be mapped back to the ensemble reference genome. And rather than what most of people would have done, including myself, and just gone with this and said, well, you only use what you can map back to ensemble. He and another talented, actually Europe student, Alex Finkel, actually devised a program that could, a new feature annotation program that could basically define what a peak was, you know, from basic principles. And this is called RNA Express. It's published, they published it in Bioinformatics, and it's available on the website for a whole range of applications, which I would recommend. But this enabled us to identify unusual features that were unexpected. And one of the main things that we found was a whole range of changes, interferon-induced changes in untranslated regions. So here you can see, so there are genes like STAT2 which have a longer 3' UTR than mapped in ensemble, you can see from the reads, and others like MMP12 here which are much shorter than that annotated. So of course this is really important for predicting microRNA targets and was unexpected. The other thing to say is over 30% of the genes are induced by interferons have this subjected to them. So they either have longer or shorter UTRs than we predicted. So everything had the expected UTRs in the uninduced state, I should emphasise. So basically, some of these we've validated. This shows TLR4 on the top, TLR2 on the bottom, which are interferon-regulated genes, so you can devise PCRs to regulate, well, at least to identify the longer transcripts. The shorter you can't by PCR anyway, because you'd be positive for both of them. And so this may in part explain, this messy slide that I just want you to run your eye down the right-hand side, in part explain at least why the ability to predict microRNA targets is so difficult, because if we haven't even got the right substrate there in the first place, then they're certainly going to be inaccurate. So that leads us then on to the small RNA and the microRNAs. So you can see here, again, over a time course, sorry, that's 0, 1 and 3 hours, you get small RNAs that are either suppressed, sorry, this is 1 hour, 3 hour and 12 hours in this case, that are either induced or suppressed. The time course vary. We've validated many of these, both induced ones and suppressed ones over a time course. Sorry, I've put the wrong data down down this side for some of it, but the sorry, that's the wrong. Basically, what I should say in summary, I'll put that's for the large RNA, not the small RNA. So looking at this, basically there are numerous categories of small RNAs, which is less than 200 nucleotides, that we couldn't classify. There are about 40 microRNAs that had already been annotated that were induced, about a similar number that were suppressed, and about an equal number, 40 induced, 40 suppressed-ish, that were newly annotated and previously undescribed. So the way Sam devised to annotate novel microRNAs was basically use the RNA express feature annotation that I just told you about, MIMR-PHI and Dario, so some of the publicly available programs, and also the requirement that there had to be a detectable precursor in the large RNA to doubly ensure it. So this gave us some improvement, we believe, in the ability to predict microRNA over that data that we just showed you. However, not absolutely perfect. So we then, in collaboration with Kate Jeffrey at Harvard and Nina Papavasilio at the Rockefeller, did a hits clip experiment, and basically what this is essentially is the equivalent of a chromatin IP if you're looking at promoter analysis. So what it does is you UV cross-link microRNAs in the RISC complex bound to their targets. You RNAs digest, you chop them up, so again, similar to a chip assay, pull down with a pan-Argonaut antibody and basically amplify up and analyze, resequence and do your bioinformatic analysis. So out of that, essentially what we got were 3,800 large transcripts, so targets, and 385 small microRNAs. If we overlap the RNA-seq predictions that Sam has divided with the microRNAs then that were validated in this hits clip procedure, you can see the overlap is pretty good, but you know, probably up around 50 to 60%. But at least what we have in this common field are basically 64 known microRNAs that are interferon regulated and 55 novel microRNAs, you know, validated by hitting a target in this system. And probably about an equal number of these are induced or suppressed. So this we were very excited about. There's probably only, you know, a dozen or so microRNAs known to be interferon regulated and impact on the system. So this we think is very important. We wanted to validate then whether the consequence of the changes in UTRs had any value in microRNA targeting. So down the left-hand side here is STAT1. So in the uninduced state, it has the longer UTR, which contains a MIR-155 site. So you can see in a reporter assay, it's active. It suppresses the transcription in this reporter. But in the induced state, where it has the shorter UTR, and you lose this, and again, you lose the regulation. Conversely, with TLR4, which has the shorter form in the uninduced state, there's no regulation. Where you get the longer form, you do get regulation. So basically, this is saying, at least in the reporter assay validation, that changes in these UTRs do have some targeting consequences for the microRNA. I mean, in this system, we're interested in finding, you know, important nodes. So situations where one microRNA might target multiple members, particularly if they're in a particular pathway, and there are examples of this, or when lots of microRNA might hone in and target one sort of key target gene, and there are examples of this as well. So just to expand on the regulation of some of these pathways we're interested in. So basically, what this graph shows is the open bars there show the targeting of the shorter form, the black bars, the longer form, and there are three different types of validation. One is a HEC reporter assay where we put in a microRNA mimic. The other is immortalized macrophages where we put a different reporter, more real cells like very much like the primary BMMs, where we again put in a microRNA mimic, or where we put in an integument. So basically what you can see here is the longer forms do show targeting by multiple different microRNAs in both these systems. And when we block it, you get the microRNAs, you get the expected increase in activity. We've also validated this for SOX1, which again shows similar targeting in each three of these assays. And I said this is something that's been traditionally difficult to modulate. So basically, in essence, we have an awesome amount of data that you can see depicted here on this wonderful summary slide. So down here are the 1,500 genes regulated by interferon. Across here are the 150-odd microRNAs that regulated. There is a sort of colour key. I think I tried to expand it, but it's not much better. But basically what you can see, so basically you can hone in here on areas of interest. So here you can see a group of microRNAs that essentially target one gene, or vertical lines show, you know, a microRNA that will target multiple genes, and where kind of working through this to try if we can see if we can find some of the really important areas of regulation. But I think this is a fantastic source of data for experiments for regulating interferon. Remember, interferon is an important part of all your pattern recognition receptor responses, and so this situation of both this targeting and the modulation of the UTRs, I think, is something that will be common to a lot of systems we look at. So I'll just close and summarise then. So basically, you know, 3 stories, one on some notions on what drives what interferon might be selected in different situations. I think some ideas that not all interferons act the same, you know, with these components of the receptor, and maybe differential interaction with these may determine different types of signalling that can occur through the system. And apart from the transcriptional regulation, which many of us have been worked at for a while by upstream STAT transcription factors and so forth, there's this complex network of microRNAs that will, I think, be important regulators and potentially modulators that we can translate of this system. So it just remains for me to acknowledge the people who've done the work. So the interferon epsilon work was very much driven by Neve Mangan and Kai-Yi Fung. Kai-Yi's now doing a postdoc with Liz Hartland. Nikki DeWeerd, in collaboration with Jamie Ross-John's group, drove the interferon beta IFNAR-1 story. And Sam Forster, you know, did a huge majority of the work, as I said, both the bioinformatics, writing the scripts and doing the wet lab validations on the next-gen sequencing experiment. I mean, a lot of my work is done in collaboration with the other lab heads in our centre, Brennan Jenkins, Ash Mansell, Richard Ferrero, who did his sabbatical here last year, travelled a long way, Phil and Carl. In the Epsilon work, we had a great collaboration with Phil Hansborough from Newcastle on the chlamydia. Dan Carr helped us set up the virus systems. Sarah on some endocrinology advice. Across the projects, we have a good structural collaboration going with Jamie's group, etc. And as I said, the HITS clip, which is a really difficult technique to get going, you know, wouldn't have been done without sort of Kate and Nina in the US. So thanks very much for your attention. And I should advertise our conference. I hope you're all coming at the end of October. Not too late to sign up, yeah.
We've got time for some questions.
I was just curious, I saw one of the earlier slides with your clustering showing the interferon alphas sort of don't necessarily line up mouse and the whole side.
Yeah.
And that's for the protein sequence. What about the promoter sequences of those various genes? Do they sort of fall? Do you find that the subtypes of the genes...
That's a really good question, and I should have done it, but I don't think we have, actually.
You would expect that if you're right, that you will get human and mouse lining up.
Good idea.
Maybe I misread that, but did you have a group with what you would accept the responsibility interferon epsilon?
Yeah, maybe. Yeah, well, yes, that's what was written there. My impression from Transfac, I think that prediction is that, you know, it's very hard to distinguish those from classic hormone receptors or progesterone receptors. So.
Well, they are dual use.
Right.
So that could be a progesterone receptor.
Yeah, we've looked at glucocorticoid regulation and we don't see any evidence of it. But
progesterone.
Yeah. Well, we again, if we look at, we've set up systems with primary endometrial cell cultures, and we don't get direct regulation by any of those hormones. So we're kind of thinking that might be indirect through a stromal cell or something, but it's hard to kind of nail the systems. I'm not sure we've got it working perfectly yet.
Well, it seems counterintuitive that you will reduce the protection conferred by epsilon interferon during pregnancy. Then, and I'm wondering if, because there is no, you are saying that there is no effect on the viability of the embryo, but I wonder...
No effect of the absence.
Is there?
No effect of the absence of epsilon on, right.
How do you try to push it? So what for example, if you across them to genetically disparate animals?
Yeah, so we're making a transgenic at the moment where we can regulate it and keep it on. So it's a good point.
Can you make an speculation of why would you reduce the expression of epsilon interferon?
Well, it's not just epsilon. So there are a lot of pro-inflammatory cytokines are suppressed at exactly that point. So there's a general feeling that there is an immune suppression, commensurate with implantation.
Really? I was wondering if you talk about the role of insulin in HPV infection and whether it plays a role in cervical cancer.
Great question. It's something we're thinking about doing but haven't done anything on yet. I mean, one thing I should say is that there does seem to be a gradient of expression. It's higher in the upper reproductive tract, lower in the lower reproductive tract, but the HSV, like HPV, is more infectious squamous epithelium, so we do still seem to get an impact. So I think it's a good question to look at.
With the interferon beta signalling just through the IFNAR1 receptor, have you tried putting an IFNAR2 soluble domain, and does that change the pattern of expression of the genes that are controlled through IFNAR1 alone?
Yes and no. So we have done that quite a while ago. So we haven't done it more recently that we've got the microarray set up to look at the effect of that signal. But that does work in a trans-signalling sense, like the IL-6 soluble receptor with GP130. But again, it's not a full response, so it's something we're sort of doing in parallel.
For the hormonal regulation of interferon epsilon, how did you measure or quantify interferon epsilon relative to the number of endometrial cells? Because they fluctuate in a very similar pattern in the ovarian and reproductive cycle to...
Yeah, good question. So at the moment they're sort of, well, I can say three things. One is they're just standardised to like total RNA loading, so that's one thing. The second thing is we've done it on sort of cultured cells and looked at it that way. And the third thing is I didn't show you, but we have immunohistochemistry, so you do seem to get less per cell rather than just a change in the number of cells.
Okay, one last question, I think, yeah.
With the IFNAR1 signalling through interferon beta, does it still require TIC2?
Yeah, don't know. Good question. Well, I don't know, because what's thought about TIC2, it might have stats as targets or the other receptor as targets, so we don't get stat signalling, we don't have the other receptor in there, so I'm not sure.
Can you see activation or have you looked for activation of TIC2 by phosphorylation?
No, I haven't looked at that. The one thing we've looked at, there is a sort of pretty well characterised alternative signalling pathway through ACT, so we do get ACT phosphorylation in that response. But it may well be stat independent. But yeah, good thing to look for.
Okay, it remains to thank Paul for a very interesting